Okay, so welcome to this next video in which we are discussing pathological cardiac hypertrophy. So we're just having a bit of a revision now of the mechanism by which beta-1 stimulation causes an increase in the force of contraction of a cardiomyocyte. I just can't resist going into the full pathway now, so we're doing this. Right, and this, by the way, this entire pathway is disrupted in hypertrophied cardiomyocytes. So this will be useful for upcoming videos where we'll see exactly which portions of this pathway are disturbed. Okay, right. So troponin T we've discussed so far, which is the troponin protein which actually binds to the tropomyosin. In between, in the middle, you have troponin C, which I've just drawn in turquoise. So this is troponin C. And then finally, the troponin protein which actually binds to the actin filament, this is troponin I. And I'll colour that in orange in a moment. So troponin I is the one that binds to the actin filament. Right, so in order to activate contraction of a sarcomere, you have to bind calcium to troponin C. So when calcium goes up, it binds to troponin C. Troponin complex then changes its conformation. It moves the tropomyosin and exposes the binding sites on the actin filament for the myosin filament. So the myosin filament can then start climbing up the actin filament and the sarcomere will contract. Okay, now, how does protein kinase A affect this process? Well, basically, protein kinase A can phosphorylate troponin I. Okay, and when it phosphorylates troponin I, it increases the sensitivity of this troponin complex to calcium. So, for a certain concentration of calcium in the cytoplasm of the cell, you will get the activation of a greater number of sarcomeres once the troponin I protein has been phosphorylated by protein kinase A. So it increases the sensitivity for calcium of the activation process of sarcomeres. Right, okay, so those are the the two ways really, by which the beta-1 stimulation activates the force of contraction of these cardiomyocytes. Basically, you're going to increase the size of the calcium spikes, which is going to mean that we recruit a greater number of sarcomeres within the cardiomyocyte, okay, which is going to lead to a greater force of contraction. In addition, what we're going to do is we're going to increase the sensitivity of the sarcomeres, and again that's going to mean that the number of sarcomeres we recruit for a certain calcium spike is going to be greater. Now, I just want to go into protein kinase A in a bit more detail. So I just sort of gave you the summary of what it does without dwindling with the mechanisms, but I now want to just discuss the two types of protein kinase A in a bit more detail. So basically, there isn't just one type of protein kinase A, pKa. There are two types of protein kinase A, cunningly named protein kinase A type 1 and protein kinase A type 2. So you have type 1 and type 2, and the difference between type 1 protein kinase A and type 2 protein kinase A is that type 1 protein kinase A is free within the cytoplasm of the cell, and type 2 protein kinase A is bound to proteins at the plasma membrane known as A kinase anchoring proteins, which we'll talk about again in a moment. Right, so they all have the basic same structure, which I'll draw here. They exist as a complex, so protein kinase A isn't just one protein, it's far more complicated than that. Instead, you have two types of protein that are assembled into a tetramer, basically. So this is the structure of a protein kinase A molecule. Okay? Right, so you have these two proteins here that are identical, basically, and these are regulatory subunits of protein kinase A. These are not active enzymes in themselves, but they are still considered part of the protein kinase A enzyme. So regulatory subunits of protein kinase A. Now, the regulatory subunits, each of these regulatory subunits, which we've dimerized together here, there are 2 genes in the human genome which you can use to make these regulatory subunits of protein kinase A. There is the R1 regulatory subunit, and if you use the R1 regulatory subunit in these 2 positions, then you will make a type 1 protein kinase A, which will be free in the cytoplasm of the cell. There is also the type 2 regulatory subunit, the R2 protein, which if you use that in these 2 positions, you will get a type 2 protein kinase A, which will attach to ACAPs, or A kinase anchoring proteins. Okay, now, these regulatory subunits don't just assemble with each other, they also assemble with 2 other proteins, which are the catalytic subunits of protein kinase A. So, you form this complex basically consisting of 2 regulatory subunits of protein kinase A and then 2 catalytic subunits of protein kinase A. And this is what's known as the R2C2 complex, because you've got 2 regulatory subunits and 2 catalytic subunits. Okay, so R2C2 complex. So this whole thing is protein kinase A, not just the catalytic subunit. Don't be fooled into thinking that this is the protein kinase A. The whole thing is protein kinase A. Now, you have these 2 types of protein kinase A, the type 1 and the type 2. These are the free ones that you just have this sort of a structure floating around in the cytoplasm. And the type 2s, you have this attached to an A kinase anchoring protein. Now, before we discuss the A kinase anchoring proteins, let's just discuss how you activate this, this protein kinase A complex. So basically, it is these catalytic subunits here that will actually phosphorylate proteins. However, they are inactive at the moment. They are bound to the regulatory subunit, and when they're bound to the regulatory subunit, they are not active. So, in order to activate them, what you have to do is bind cyclic AMP to the regulatory subunit. And cyclic AMP binds in these little invaginations that I've left here, basically. So you can bind a cyclic AMP there, a cyclic AMP there, okay? And I realise that it's gone a little bit squashed. But that's just to fit it onto the page. So basically what happens is that 4 cyclic AMP molecules are going to bind to our regulatory subunits like so. So these 4 pink dots here, these are cyclic AMP molecules. So one of these binds to each of these cyclic AMP binding sites here. Okay, and when that happens, what's going to happen is that the complex, well, these regulatory subunits are going to change conformation basically, and when they change conformation, they're going to release these catalytic subunits, which I've now drawn in blue. So the catalytic subunits are now going to be released, and once they've been released, each one of them is a functional serine-threonine kinase, which is going to add phosphate groups onto the serine and threonine residues within proteins. Okay, right. So, these are then going to go off and phosphorylate their targets. So, that's how you activate both forms of protein kinase A. So, let's now talk about how the type 2 protein kinase A's are different to the type 1 protein kinase A's. So, the type 2 protein kinase A's will bind, basically, to proteins known as ACAPs, which bind to other proteins in the plasma membrane. So, this here, this is supposed to represent an ACAP, okay, and we'll have it here, and it's bound to some other protein that's maybe in the membrane up there. Right, so ACAP stands for A kinase anchoring protein, and A kinase is basically just shorthand for protein kinase A. So this stands for the A kinase anchoring protein. Okay, and it's called that because it is going to bind protein kinase A to the plasma membrane. Well, it's going to bind protein kinase to it, but then it's bound to the plasma membrane, so effectively it's going to anchor the protein kinase of the type 2. So it's only protein kinase A's which have the type 2 regulatory subunit. Okay, so that's the difference between these two. The catalytic subunits in both types of protein kinase A are utterly identical. It's the regulatory subunit that changes. So this is the type 2 regulatory subunit here. Okay, and it's got those catalytic subunits bound. So the type 2 regulatory subunits will bind to A kinase anchoring proteins, whereas the type 1 regulatory subunits won't, so they remain free in the cytoplasm, basically. So if I draw another one of these free in the cytoplasm, this is one where both of these regulatory subunits, here's one regulatory subunit, here's the next one, these are both type 1 regulatory subunits now. Okay, right, so that's the two types of protein kinase A. Now, basically, which types of protein kinase A are going to do each of these activities that I have explained to you? Well, basically, in the case of phosphorylating troponin I, this pathway that I've just talked about, and also in the case of phosphorylating SERCA, it's the type 1 protein kinase A. So basically, when you activate the beta-1 adrenergic receptor, it's going to activate the production of cyclic AMP by adenylyl cyclase 5 and 6, and this is then going to activate free protein kinase of the type 1 in the cytoplasm, which will then phosphorylate SERCA. And specifically, I should have said this earlier actually, there are different forms of SERCA, and the form of SERCA in the heart is SERCA2, okay, and it's also going to activate troponin I. So don't worry too much about this too. It's just there are different genes coding for SERCA pumps, and the specific gene that is found within cardiomyocytes is SERCA2. Right, now let's discuss the L-type voltage-gated calcium channels and also the type 2 ranitidine receptors, because those are activated by these type 2 protein kinase A's. Now, what's quite nice about this is that you form these beautiful complexes involving A caps. Okay, so let's discuss this then. So, what basically you'll have is an A cap protein attached to the plasma membrane, which will have a type 2 protein kinase A attached to it, and then it will also bind to the target, basically. So you have an A cap which will bind to the type 2 ranitidine receptor. So let me show you this. So basically, what happens is that you form these beautiful complexes. So if this is the plasma membrane here, okay, plasma membrane's here, then if this is the sarcoplasmic reticulum membrane here, so this is a calcium synapse basically, then let's say this is the type 2 ranitidine receptor here, okay, then basically if we put our adenylyl cyclase enzyme here, the A cap will bind to the adenylyl cyclase enzyme. It will also bind to the type 2 ranitidine receptor down here, so this is the RYR2, and it will also bind to protein kinase A of the second type. So basically here comes this A cap, which is going to form a complex with all of them. And this is what's known as MA cap beta. Okay, so this is MA cap beta, and that stands for muscle-specific A kinase anchoring protein beta. And specifically, the adenylyl cyclase form that it complexes with is the adenylyl cyclase 5. So remember, we had these two forms of adenylyl cyclase in the heart, adenylyl cyclase 5 and adenylyl cyclase 6. But the one it specifically likes to form complexes with is this adenylyl cyclase 5 form. Okay, so it binds to the adenylyl cyclase 5, which will produce cyclic AMP when activated by the alpha-S GTP subunit, and it also binds to this type 2 ranitidine receptor down here. And this A cap will also have the type 2 protein kinase A bound to it, which I'll have to sort of squash in over here now. So here's the type 2 protein kinase A, rather squashed, but you'll get the message. Okay, so here's this type 2 protein kinase A. So those are the two regulatory subunits of the second type and the two catalytic subunits there. So the idea is that you've got all of the components of this signaling pathway very close by. So the adenylyl cyclase will produce the cyclic AMP, which will activate the type 2 protein kinase A, which will then go and phosphorylate the type 2 ranitidine receptor. And that will then raise the conductance for this type 2 ranitidine receptor, and also raise the threshold for inhibition. Okay. Now, a similar complex exists for the L-type voltage-gated calcium channel, which we'll see in the next video.